Hi, everyone, and welcome to our fifth web seminar celebrating research on aging. I'm Chris Rebelo, the Director of Communications here at the Buck Institute, and I'll be moderating today's conversation with Dr. Stephen Austad, who is in Birmingham, Alabama, at the University of Alabama. This is our fifth interview in our series of 6 programs involving experts who participated in a very successful symposium here at the Buck last December. That symposium celebrated 30 years of research on aging. These seminars are interactive. As moderator, it's great fun to include your questions in these conversations, so please take note. There's a chat feature on your screen. Please use it if you'd like to pose a question. Now it is my distinct pleasure to welcome Dr. Austad to our program. He is the director of the Nathan Schoch Center of Excellence in the Basic Biology of Aging at the University of Alabama. He specializes in comparative biology and aging, and he's also active in efforts to study existing human medicines to see if they could help all of us live better longer. So we have a lot to talk about. Steve, let's get right to it. Let's talk about comparative biology and aging. What does that involve?
Yeah, so this is a really major point, and I think it's actually a point that many of the researchers in the field miss, and that is all of the traditional animals that we use to study aging are demonstrably really terrible at it. They fall apart in a hurry. They have absolutely no defenses against those destructive processes that go on inside of us. The idea is that if we can figure out how those processes work in those animals that don't do it very well, if we can make them better, that will provide us clues to be able to make our own processes better. However, there's a big sort of act of faith in that. The idea is that we already live a long time. We're already pretty good at combating the features of aging. So the goal of comparative biology is to identify and study species that evolution has produced that actually do things better than we do, to basically use them as a model. One of my favorite quotes is that evolution is smarter than you are. And I really believe that evolution has had billions of years and trillions of individuals to which to try out different ways to slow or prevent aging processes. And so what comparative biology does is it acts like prospectors out there in the natural world. Can we find species from which we can learn key lessons that might escape us if we only studied the short-lived animals, which is what most of the traditional laboratory animals are.
So pick a long-lived animal that you enjoy studying and tell us what there is to learn from those animals.
Sure. So the one that I've done the most work on is a clam. And it's a clam that lives over 500 years. And I have to tell you that I'm not a clam expert at all. I got involved on this completely randomly. Some oceanographers called me up out of the blue and said, we work on a clam that lives a really long time. Would you like to collaborate? And so I said, well, what do you mean by a long time? And they said, 500 years. And I said, I'm sorry, I thought you said 500 years. And they said, no, we did say 500 years. So we started a collaboration. And what we've learned from that collaboration is that these clams, and maybe other things that are exceptionally long-lived, are very, very good at preventing their proteins inside their cells and holding their bodies together from misfolding and from aggregating. And that just sounds like such a simple thing. But if you think about it, all the chemical reactions inside our body work because of proteins, and those proteins are ones that make those chemical reactions go. But for them to work, they have to have a very precise folding. If you saw a picture of a properly folded protein, it would look like the most complicated origami that you had ever seen in your life. But over time, proteins get attacked by various things, and they gradually start to lose that fine folding that they need. And when they do that, the sticky parts of proteins start showing up on the surface. And so they'll stick to other proteins. And the next thing you know, you have a glob, you have a mass, you have a plaque if it's in the brain, and we call those Alzheimer's plaques. And we know that a number of diseases of the brain of aging are actually associated with these misfolded proteins. But I think personally, that's just the edge of the wedge. I think that misfolded proteins are part of the problem of virtually all the problems that go on inside our cells and help our cells communicate. So from the study of this one clam, we've discovered they have something in their tissues. And unfortunately, we haven't discovered what that something is yet. But we know that it helps prevent proteins from aggregating. And it helps clam proteins, keeps them from aggregating. Who cares about clam proteins? Most of us don't. But it also prevents our own proteins. And we've taken what I now think of as the Alzheimer's protein, known as A beta, and we've tried to aggregate that in basically clam juice. And it's very, very difficult to do. Much more difficult than is if we put it in a similar solution that we took from human tissues. So that's something it's a natural product. We don't know what it is, but it's not surprising. And there may be many other things that we can learn from long-lived organisms like the clam.
And so the ultimate goal is to develop therapies that would build on the success of those long-lived animals?
Yes. So the idea is we already do this to a certain extent. In the 1970s and 80s, pharmaceutical companies spent quite a bit of money sending people out in the rainforests and diving down in the ocean to discover natural products that would fight cancer. And they were really quite successful. In fact, most of our cancer pharmacopoeia today comes from chemicals that were either natural products or are modified forms of natural products. So for some reason, though, they didn't tend to do this when it came to aging. They only fixated on cancer. But I think it's time. Now, some of the molecules that you've heard of in aging are also from natural products. Resveratrol, for instance, has gotten a lot of publicity in the aging world. That's also a natural product. So I think we haven't yet begun, and then some of the anti-senolytics that we may talk about at some point are also natural products. So it's not surprising. There's lots of reasons that plants and animals in the world would want to resist the damaging processes of aging and would come up with novel ways to do it. You know, we've been separated from plants for, several billion years. So the fact that they might have some different solutions to the problems is not surprising. We can learn from them. We can learn from the natural world.
So let's talk about big animals. So whales. So that's whales and elephants. I think when we talked earlier, you said they particularly have an ability to resist getting cancer?
Yes. So this is a really interesting feature of the natural world that I think relatively few people appreciate. And that is this: assume, which is something that's true, that any cell in your body could potentially transform, turn into a cancer, and ultimately kill you. Well, if we have about 30,000 more cells than a mouse does. So that means that each of our cells has to be at least 30,000 times better at resisting turning into a cancer than a mouse cell. And that's only if we were going to live 2 to 3 years like a mouse. So we already have much, much better mechanisms protecting us against cancer than mouse. Now think of a whale that has 1,000 times more cells than we do. They've got to be even better at preventing their cells from transforming into cancer cells, or an elephant. An elephant of course, has many, many more cells. And so even if they only lived as long as we do, they'd need to have better cancer protection. For elephants, we've started to figure out how they do it. It turns out that one of the genes that protects us from cancer, that tends to either kill a cell that's in danger of becoming cancerous, or else not allow it to reproduce anymore, that gene we have one copy of. Elephants have 19 copies of it. So a lot of our cancers, that gene has been disabled by a mutation. But for an elephant, it would have to disable many, many, many of the genes. And since mutations occur randomly, the chances that you're going to get all those things mutated and make them dysfunctional is much, much lower. So we know how that works for elephants. We're less sure of how it works for whales. So even whales that don't live any longer than we do, we have a lot to learn from. And the same from elephants, surprisingly.
So naked mole rats are a big of interest in the research on aging. So they're rodents, bigger than mice, but they're rodents. They live like what, 30 years or something, and they're resistant to cancer as well. What do we know anything about that?
Yeah, so that's a really good example. So here's something, a naked mole rat, it's about the size of a mouse. It's actually very similar in size. And it lives roughly 10 times longer than a mouse. So that's pretty incredible. But what's more incredible is that mice mostly die of cancer. If you had one thing to choose to say, this is what mice die of, it's cancer. Naked mole rats, it's very rare. There's only been a handful of cases of cancer even reported in naked mole rats. Now, we haven't nailed this down the same way that we have the elephants, but there's some really good indications that they make a certain kind of molecule, hyaluronic acid. We make it as well. It's in your skin. They sell it in the beauty stores as well for skin creams and all. But they make a specific kind of it that's got a much higher molecular weight than the kind that we produce. And there's some indications that that may be behind their tremendous cancer resistance that they have. So again, here's an animal, it came into the scientific world. It's got a really unique social system. That's why it first got attention. But then it turned out it had all these other features, and now it's really at the forefront of a lot of really excellent aging research, a lot of which is going on right there in California with Dr. Buffenstein from Calico.
So I heard that you're really interested in bats. What's the deal with bats?
I am interested in bats. Bats are the neatest animal that nobody knows about. I have to tell you that a few years ago, I was river rafting on the Snake River up in Idaho, and I was sitting there talking to the guides about bats and how neat bats were. Now, these are guides that spend the whole summer on the river. And they said, that's really interesting, but we've never actually seen a bat. And I said, really? There's one right there. And it turns out that there were all these things fluttering around at dusk they assumed were birds. Turned out they were bats. So here's the incredible thing about bats. First of all, they're exceptionally long-lived. They're the longest-lived mammal that we know of if you correct for body size, for their body size. So a little bat that's a quarter or a fifth the size of a mouse can live 30 or 40 years. And here's what makes it even more amazing. It can live 30 or 40 years in the wild. When we say that naked mole rats live 30 years, we mean, well, in the laboratory where we can protect them from a lot of things. But bats live 30 or 40 years in the wild where they have to deal with temperature extremes, they have to deal with storms, they have to deal with predators, they have to deal with pathogens, they have to deal with all this stuff. And despite that, they have this incredible longevity. So the question is, how do they do it? And this actually, I haven't even begun to scrape the surface of the things that they do that's so amazing. If you or I break a leg, get in a car accident, we spend a week or 2 weeks in bed. When we get up, our muscles have shrunk. It's hard to walk. We can barely get around. Our respiratory system needs a little bit of work. Bats, on the other hand, can hibernate. They can hang there in a cave for 6 or 8 months, and they wake up and they fly away. Now, just think about that. Flight is one of the most energetically demanding forms of transportation that we know of. And so they've got this incredible ability for their muscles to stay relatively inactive for month after month, and yet not degenerate to the point where they can't do something really energetically costly like flying. The other thing that's neat about bats is their high frequency hearing. Now, you might not remember how they hunt, but the way they hunt is they scream and then they listen for the echo, and they use that echo to locate their prey. It's called echolocation. Well, their high frequency hearing is critical to their survival. If they lose that, they're dead. They can't hunt. our high frequency hearing starts to deteriorate when we're 12 years old. You know, when my kids were in high school, they used to torture their high school teacher because they had this cell phone ring that they could hear, high school teacher could not hear. And that's because we start losing our hearing in about a decade and a half. But bats maintain it for decade after decade after decade.
So, go ahead.
Yeah, so you put all these things together, their exceptionally long life, their ability to preserve their muscle function, their ability to preserve their hearing. And this is an animal that I think we could learn a tremendous amount about how to age successfully.
So I was going to ask this question later, but I'm going to ask it now. How did you get so interested in other animals and how they age?
Well, I have a pretty unusual background for someone in this field. My PhD was in evolutionary ecology, and I was a field biologist. And I got interested in aging because I discovered some animals that aged exceptionally fast. I was studying opossums for a completely unrelated reason, and I discovered that an opossum, very much like the ones you have there in California and we have here in Alabama, only live about as long as a mouse. And I was astonished at that. I couldn't believe that. And I went out into the literature to see if maybe I was wrong, and it turned out, no, I was right. And that's what led me into my interest in aging. But because I had a background, you know, I trained animals for the movie business, I trained big cats, but I studied birds in graduate school, I studied spiders in graduate school. I sort of had a exposure to a lot of different species. And so I'm always thinking very broadly about animals that we can learn from. And so, yeah, I think that's why I come out of nowhere with these bizarre ideas. Well, let's look at bats, or let's look at opossums. You know, my first work in aging biology was working on opossums. And so nobody's followed up on that work, by the way. So I guess if anybody ever will, it's going to have to be me.
So that's a lead into a question we got from a participant here. Can you tell us about your problem-solving process in aging? As sort of a maverick, you have tackled some issues in aging different than your colleagues. What is an OSTAT approach to problems in aging?
Yes, that's a very good question. I think the way I've gone about it so far is the first thing I've done when I've come up with a species that I think we might be able to learn something new from is to see if we could culture the cells, if we could grow the cells in a dish and maybe learn something from the cells. Now, sometimes you can't do that, and there's only a limited amount of things you can learn from studying cells in a dish. That's certainly a way to start. Now, when it came to the clams, it was particularly a problem because no one had been able to really culture their cells for any length of time in a dish. So then we had to think about, well, what is it? What's the most critical thing that a clam would have to preserve in order to live 500 years? And we thought, well, it's going to have to avoid cancer. Okay, well, cancer is hard to study. Well, it's going to have to be able to preserve that muscle that opens and closes the shell. I said, let's think about that muscle. What could we possibly learn about that muscle? Well, we thought about mitochondria, because that's the energy source inside your cell. That's key to energy. We thought about cell replacement. Are there stem cells in those? And then we thought, because there's a kind of muscular dystrophy that comes from these misfolded proteins aggregating. We thought, well, let's just take a look at that. And we came up with a very simple experiment. You know, one of the things I try to do is try to think the simplest experiment you can learn something from. And that simple experiment was something like, let's put a protein that we can follow in basically clam juice, see if it resists folding. And we come up with a way to really make it fold. There's various ways that you can change the acidity, you can add things that will tend to make it misfold, and let's see if it works, if it's harder to misfold in the clam. And so that's how we came up with that. With the bats, we started, again, working on cells. And then we started looking at DNA damage. We think resistance to DNA damage is a key for the bats. The bats, fortunately, I had a colony, a couple of million bats when I was in Texas before I moved to Alabama. And there I could just go out and catch some, and I could bring them in the laboratory and take some blood, take them back, release them again, and do quite a bit just from their blood. We actually put some we irradiated their blood so we could break their DNA, and then we could look to see how well that DNA repaired itself. And it repairs itself very, very well, as you might expect it. So there's not an overall approach. There's something that just makes me, the first thing I try to do is I really try to think about the detailed natural history of the animal and what might it be particularly resistant to, what aspect of aging. You know, is it something about preserving the muscle, preserving the eye, preserving the hearing, preserving everything? For the clam, I thought it was the muscle.
So before everybody runs out and drinks clam juice because it's good for the clams, do you have anything to say about that?
One thing So my wife wants me to start a company selling the elixir of clam immortality juice. So far, I've managed to avoid any attempt to commercialize this stuff, but I even have a name for it, and you're welcome to that name, Clamour. Please don't. You know, I tell people all the time, one of the questions I always get, well, what do you do? What do you do to stay young and healthy? And I say, well, I'm not a mouse, so I don't do A, I don't do B, I don't do C, just because we know it does well in a mouse. I don't drink clam juice. In fact, I have to tell you, I used to love clam chowder. Now when I'm eating clam chowder, I'm thinking, that could be 500 years old, that clam that I'm eating. I don't really enjoy it the same way I did before.
That's great. So let's move on to another area that you're really interested in, which is sex differences among species, among humans, involving aging. Talk about that focus in your research.
Yeah, so this is something that I've come to in the past 5 or 6 years, which is, I mean, it's obvious if you're a human that men and women age in a different way. Women live longer. There's a handful of diseases that they are more likely to get than men, mostly autoimmune diseases. But for the most part, women are more resistant to almost all the major causes of death: cancer, heart disease, infectious diseases, you name it. I always thought of that as a kind of an interesting feature of human biology, but not something that I really thought worth studying. I thought, well, when we come up with some therapies for slowing aging, it's not going to matter if you're a man or a woman. But a couple of things happen. The first thing that happened is people started doing mutations in mice to make them live longer, genetic mutations. And sometimes those mutations would only make the males live longer. And other times they would only make the females live longer. And they would only keep the males healthy longer and only make the females. Now, both sexes are having the same gene knocked out. So there's got to be some underlying bit of biology. So that's when I really started thinking about it. And then when people started giving drugs to animals, particularly the National Institute on Aging's Interventions Testing Program, it turned out that the drugs they started to identify either only increased the longevity and health of one sex or increased it much more in one sex than the other. And so I thought that there's really a deep message here about aging biology that we're not paying sufficient attention to. And so now I'm actually using this difference in the effect of treatments on males and females to try to understand more about how aging proceeds differently. Now, at my university and many universities, there's a real focus now on personalized medicine. Not all medicines are going to work the way they work in some people, in all people. It strikes me that this is the first and crudest pass at personalized medicine. Are we going to have medications for women and medications for men that are different. And we haven't studied this a lot in the clinical community. There's very few drugs that we say, oh yes, these work for women, but not for men, or vice versa. But there's some really interesting work coming out now, particularly in the pain field, where they know there's certain kinds of pain that you can give a mouse and you can give a drug that will ameliorate that pain to a male, but will have no impact on the female. You need to use a different kind of drug to make that ameliorate that pain in both sexes. So I actually think that we're on our way to using what we're learning from aging to maybe revisit not just aging, but many of the ways that we treat diseases in the clinic to see if women and men might do better by having slightly different treatment regimens. I think that's a really intriguing way for medicine to make up for all the years that they only studied men, basically. Right. And now it's mandated that you can't do that. It's been a very good thing for the field that it has been mandated.
So isn't it rapamycin that had different results in genders? Was there something with that?
That's one of the drugs, and that's a particularly interesting one because it seems to have at the lowest doses. This is in mice now. Let me emphasize this is in mice. At the lowest doses, it seems to extend life much more in women, much more in females than it does in males. As you increase the dose, the effect gets more and more similar, and eventually you reach a high enough dose, it doesn't help the females anymore, it only helps the males. So there it's completely dose dependent, it seems like. And so again, this again, it's teaching us something about, it's deepening our understanding of biology by looking at something that 10 years ago, we were just, it's one of these things that nobody paid any attention to. You know, if you ask aging researchers, well, women live longer than men, right? Isn't that interesting? Well, yes, but you know, what can you learn from it? Well, now that we've started to look into both sexes of animals, we're learning a lot from it. And I think we can. You know, there's a lot of diseases that are biased toward one sex or the other. Mostly they're biased towards males, but there are some that they're biased towards females. For instance, more women than men die of Alzheimer's disease, even if you control for age. Now, it's not clear that women are more susceptible to getting Alzheimer's disease, but they're more likely to die of it. That may simply be because women survive longer with it. I mean, one of the things we now know is that in times of stress, in times of serious disease or food stress or other stress, women survive better than men. Women deal with that. In fact, female babies deal with that better than male babies do. Again, it's one of these things that if you ask a neonatologist, they could always tell you that. Oh, yeah, if you have a sick baby, you really want it to be a female because it's much less likely to die than if it's a male baby.
And they still don't really know why?
No. I mean, and there, you know, the first thing we think about when we think about adults is hormones. Okay, well, males have testosterone and females have these estrogens. That must be it. But now we're talking about babies, you know, so a long time before hormones have really determined their biology. Now they do have this surge of prenatal hormones, but right after they're born, there's not a huge hormonal difference. But there's lots of other things as well. I personally am feeling like the Y chromosome hasn't gotten the respect that it's due. Tiny chromosome, it has a very few genes on it. But I think there may be some genes on the Y chromosome that we really need to pay attention to.
Great. So there's a question that is about sort of more traditional, if you want to call that, research on aging. Do you believe that yeast and fungi share certain aging mechanisms with mammals, including humans? And what can we learn from studying those simple model organisms? What's your take on that?
Well, we've already learned a lot from them. I mean, to a certain extent, the last 30 years of progress in aging biology, we can thank these simple organisms for. Now, they're not the same. Yeast is a single-celled fungus, but it's got most of the same cellular components. It's got mitochondria. It can help us identify certain gene pathways that we can then go on to the other. The interesting thing about these simple organisms is they've been separated from one another a long time too. So if we find something that works this way in yeast and works the same way in a simple worm, then those are very, very ancient pathways, and they might well be conserved in us and work in the main way with us. So I'm not a fan of mindlessly extrapolating from the simple organisms to humans. In fact, not even extrapolating from mice. Some of my colleagues think that mice are just small furry humans with a long tail. But in reality, the record of cures or treatments moving from mice to humans is not a great one. About 1 out of 10 cancer therapies that shows great promise in mice, excuse me, ever makes it to the clinic in people. In Alzheimer's disease, so far we've had over 300 successes in mouse versions of that. And none of those have translated to the clinic. So I think it's always good to be somewhat skeptical. That's why I made the comment earlier that I don't do this or that because I'm not a mouse, because I do think it's very easy to see the hope that comes out of some major breakthrough in worms or flies or in mice and say, aha, we finally figured it out. The year that this first rapamycin paper came out, 2009, I was at a conference in New York to talk about rapamycin, and this guy came up to me, he goes, I'm a stockbroker, I make a lot of money, where can I get some? And I said, whoa, wait a second. First of all, we've had one study, and it's way too early, and it was in mice, and now, of course, we've had dozens of studies, and rapamycin is looking more promising than ever, I have to say, because there's actually been some of the things that we've seen in mice, we've now seen in people. Particularly its effect on enhancing vaccine response. So I'm quite optimistic, but I do think we need to be cautious about assuming that if we see something in a worm or a fly or even a mouse, even though it's a mammal, there are 5,000 species of mammals, and it would be hard to find one that's more different from us than a mouse is. So just a note of caution. We're all optimistic. I think we're optimistic because there are so many things that we now know that we know that can enhance the health of a mouse. Some of those things I think are going to translate. The key is going to be able to be figuring out which of those do translate.
So that's a good lead into a question and comment I got from somebody who works in data and technology, and I can forward you, I think it's her email address later. But where can folks from data technology sector help both your work and the overall field of aging? This person's in a group whose expertise lies in data and artificial intelligence. Talk about, because AI is really an up and coming thing using machine learning. How might that help move the field forward?
Well, I think there's potentially a huge role that it can play. Because the way we're doing things now, I might pick something like protein folding and look at it. But there's now the technology for us to look at all the proteins in the cell, all of the metabolites in the cell, all of the messages that the DNA is transcribing in the cell. And what we're getting more and more data, but understanding exactly what we've got is getting harder and harder and harder. We're basically beginning to drown in a sea of data. I'm very familiar with that because when I was in ecology, that was the same problem we were having there. We were drowning in a sea of data, and we needed some structure to extract the real information from the data. So I think this is really where the AI people can come in and help us. Take all the omics data, the metabolomics, the proteomics, all of that stuff that's coming, the genomics, and try to understand how it's all put together to make this species stay healthy for 2 years, this species to stay healthy for 80 years, this other species to stay healthy for 500 years. I think we can really have a fruitful collaboration with the AI people.
That's great. So you are also the scientific director of the American Federation for Aging Research. I'm a huge fan of those folks. There's something called the TAME trial, or the prospective TAME trial, which focuses on the drug metformin. Talk about that, and where does that stand?
Sure. The TAME trial, TAME is an acronym. It stands for Targeting Aging with Metformin. And metformin is a really, really interesting drug to the extent that it's, first of all, it's already approved for human use. It's the #1 prescribed drug for type 2 diabetes in the world. It's been used for 60 years, so the safety profile of metformin is really, really well worked out. Now, the reason there got to be the interest in it is that some observational human studies have suggested that people taking metformin get less cancer than people not taking metformin. And other studies, and I'm not talking about one study here, I'm talking about dozens of studies. Same thing for dementia. The same thing for cardiovascular disease. And then there was a study a few years ago where they compared thousands of people taking metformin because they were diabetic. And by the way, uncontrolled diabetes mimics aging in many, many ways. So a bunch of diabetics taking metformin with a bunch of non-diabetics, and they matched them for weight and for income and for all kinds of stuff. And the people taking the metformin were living longer. The people with diabetes and metformin were living longer. So this really got a number of us interested in doing a human trial in metformin. It's called the TAME trial. And for the last few years, we've been struggling to get it funded. And now it looks like it's ready to go. I have to tell you that right now it's going to start. It's going to start soon. Within weeks, I would say we will probably be starting to recruit patients for this trial. Now, the people we want to recruit, they have to be at least 65 years old, between 65 and 80. And there are a number of things that they have to have. But this is ready to go. And there's a number of reasons that I think this is so important. The first thing is it's the first trial that will have 3 different outcomes that are clearly important. One is preventing dementia, the other is preventing cardiovascular disease, third is preventing cancers. That's a single drug. And this is something new. The FDA does not typically look at drugs for multiple disease outcomes, but they've been very receptive when we've talked to them about that. The other thing, I think if we can show that in a 5 or 6 year study, we can really determine whether a drug has successful anti-aging properties, that's going to get some of the big pharmaceutical companies more interested in this than they've been, because they've always assumed, well, you need a 50-year study and the FDA will never approve it. Well, what the TAME study is going to show is that, first of all, you can do it in a 5-year study if you design it right. We spent a lot of time on the design. And not only that, but you can get it FDA approved, assuming that it works. And so the TAME trial is, AFAR is very proud. We've been working very, very hard on this, and it looks like we finally got all the pieces in place to get it underway. So we're really excited. So stay tuned.
So, so for folks who are going to be interested in this, is there going to be a website where they're going to be able to go to see if they qualify and sign up?
There will be a website, and what I suggest that they do is put the AFAR website on bookmark the AFAR website on their browser, because that's one of the first places you'll be able to find links to the TAME trial once it's recruiting people.
That's great. So a question here. Hi, Stephen. What areas of the aging field excite you the most, both short-term and longer-term?
Well, I think in the short term, the one that I find that the most intriguing, I'd have to say, is the young blood thing. Now, I'm not involved in this at all, But for those of your listeners who may not know about this, if you transfuse blood from a young mouse into an old mouse, or even from a young human into an old mouse that's had its immune system wiped out, it has all kinds of wonderful properties on the brain, the muscle, the heart. And if this turns out to be true in humans, it sort of confirms a lot of old wives' tales. But B, as I've been telling my students here at UAB, guys, this is a business opportunity. People are looking for serum from 20-year-olds. Thousands and thousands of them here, right within a mile of me. And so, this is a solution to college debt, guys. All you need to do is sell that plasma, find somebody who wants a transfusion from you. So I think that's a particularly exciting one. I'll be interested to watch that develop. And I'll tell you, the things that excite me the most are the things that have been found by a whole bunch of different labs. Somebody can find, Richard Feynman said, well, the thing to do if you're a scientist is not be fooled, and you're the easiest person to fool. And I think, we always think that what we're doing in our own labs is the most important, but that makes us easy to fool about our own results. So, my opossum study, the study that got me into aging, I started that study because I wanted to test an idea that I thought was absolute bunk. And by golly, it turned out it wasn't. And that to me is the kind of strongest science, when somebody, preferably that hates your guts, repeats your experiment and finds exactly the same thing.
That's great. So a word of caution from a question that we got: are there any complications of getting different blood from others? So before people run out there and start finding young people to transfuse, are there some caveats?
I wouldn't suggest, remember,
Yes.
In mice, this is done in mice. So I'm not suggesting, that was a joke about my students. I wouldn't suggest you go out and find a young person to share blood with at this point. The other thing about this, by the way, is that the blood from the older mouse made the younger mouse older. So there's that too. No, there's lots of things to think about. There's blood typing. I mean, we know a lot about what kind of blood you can be transfused by and what you can't be transfused by. But let's wait for the science to get there. That's really what I want to do. Don't go out and find some charlatan, because there's somebody who'd be willing to sell that to you, I guarantee you. Wait till the science is in. This is a really active area of research. Very good scientists are working on it. Just wait. Be patient.
So back to the science, there's a question: can you comment on biomarkers of aging? What would be the best clock for which animal modeling for the human? Is there, and they asked if protein misfolding might be one. What's happening with biomarkers, and how do they fit in to the overall field?
Yeah, well, biomarkers are really important, and they're really important in two senses. A biomarker is an indicator of, in the aging field, would be an indicator of biological age that's better than what's written on your driver's license, right? And it's been a real challenge. The NIA, the National Institute on Aging, put $100 million into studying biomarkers 20 years ago, and they came up with nothing. But we've come a long way since then, and now there's a lot of stuff starting to come out. Markers on the genome, certain protein profiles. Now, the importance of these things is twofold. First of all, if we can find ones that reliably tell us something about biological age, and meaning how far in the future you can expect to remain healthy. First of all, the thing that does is it shortens our experiments. Instead of waiting 3 years for all of our mice to die, if we could, in 6 months, be giving them this drug and say, we can see they're going to be staying healthy 20% longer, then that really accelerates the pace of research. The other thing is it accelerates the way in which we can move stuff into humans. If we have good human biomarkers, we could do the same thing. Instead of us doing the TAME study for 6 years and waiting for people to get or not get heart attacks or dementia or cancers, if we could do it for 6 months and say, look, all the biomarkers tell us this is keeping these people younger, then that really increases the rate at which things can move from mice to dogs to people.
So we talked about metformin. What about the effort to really look at drugs that are already in the market that could help with aging? Maybe you're already helping with aging, but we don't really know that they do that. What's that work about?
Yeah, well, I think a lot of people are doing that. And you know, rapamycin was already in the market. Metformin's already in the market. There's another drug that in mice, male mice is showing great effect. There's one called 17-alpha estradiol. These are already in the pharmacopoeia for some things. Those are the ones to look at, I think, because first of all, they've already been approved. They've already been approved for the Food and Drug Administration for certain things. And that makes them easier to work with, and also makes it simpler to get them to get their range of approval expanded to aging. Now, the pharmaceutical companies aren't necessarily interested in those so much because if they've been around for a long time, they might be off patent, like rapamycin is off patent and metformin is off patent. But it could be what's happening with rapamycin is that the drug companies are tweaking it a bit to make it, let's say, to make it stay in the blood longer, to do things. So I think that's absolutely the thing to do is to exhaustively search existing approved drugs, because if we hit on something there, the pathway to your bathroom counter is much quicker.
Another, simple, I guess, in one sense to do that there's a lot of interest in the field is intermittent fasting.
Right.
That shows, that appears to have health benefits. And I know when we talked earlier on the phone, you talked about some of your field work. So talk about intermittent fasting and maybe some of your personal experience with it.
Sure. Well, this is a new idea. One of the things, for 50 years, we only had dietary restriction as a tool to tweak with aging. And we always assumed that for dietary restriction to have a major effect, it had to be done. We had to eat a lot less every day, month after month, week after week, year after year. But what's happened recently is people have begun to realize that a lot of the advantages of dietary restriction seem to happen quickly, seem to happen within hours. And so now there are these new diets that, again, this is not proven yet, but it's looking quite promising, where short periods of fasting, periods as short as 15 or 16 hours each day, can actually stimulate a lot of molecular processes that we think are important to maintaining health. So a lot of people, and I'm trying this right now myself, not because I think it's a miracle, just because I think it would be a good way to maybe keep weight off. Eating, I like eat in a 9-hour period each day, and I don't eat anything. But again, I'm not expecting miracles, but if miracles are going to come, then I'm welcome to accept them. Now, what you were talking about is that when I used to do field work in Papua New Guinea, and I worked in a very, very remote area of Papua New Guinea, which was a week's walk from a jungle airstrip. And the people there eat once a day. They eat once a day pretty much out of necessity. But they will wait and have all their meal basically just before dusk. And that's it. Now, these people don't live a long time, I have to tell you, because it's very, very rough life. They have lots of infectious diseases. But they don't get a lot of the diseases of aging that you see elsewhere. I never saw a single person with osteoporosis. I never saw a single person that was demented. And these days they get enough, missionary doctors coming in when they're kids to give them immunization, that some of them are living into their 70s and all. And you'd expect to see at least occasionally these dementia. Also, they have one of the highest risk factors in the world. They have a gene that's very common there called ApoE4 50% of the people in this part of New Guinea have that gene, and yet they don't report dementia. Whether this has anything to do with their diet, I have no idea whatsoever, but it's kind of an intriguing hypothesis.
Wow, so you can learn a lot from different groups of people who are in different cultures, other places. That's really wild.
Yeah. I mean, I went over there to study some animals, and I got more interested in the people than I did in the animals.
That's great. What do you see as the biggest barrier in the field, or barriers? Like, if you could wave a magic wand and change something, what needs, the field is really, I've been working in this for a while. The field is really just, it's amazing how fast it's moving. But what are some of the barriers? What do we need to tackle to really get from point A to point B to point C?
You know what I really think would help? I think what would really help is having an additional species or two that we could test treatments in before they go to humans. And I'm a huge fan for the companion dog work that's going on right now. Again, I'm not involved in that work, but I think because dogs live with us, because they get way more similar disease profile to us than rodents, that if we come up with something that looks really promising, we can test it really quickly in dogs. And the other thing is, we're not using dogs as guinea pigs here. We're really using them to try to improve and extend their health. And everybody, I think, who likes animals is into that. If for instance, the Dog Aging Project is starting this rapamycin trial, well, those dogs are getting better veterinary surveillance than anybody's else's dog. And if we find something and it had an effect in the dogs, and we know it has an effect in the mice, my optimism about it being healthful in people would just shoot through the roof. So I think if we just had a national goal of adding a couple of species that we care about, because we could be helping them as well as helping ourselves. I know for anybody who has a big dog that, lives 6 or 7 years, they would love to have something to make it live as long as a Chihuahua. I certainly would. I've got 6 dogs myself.
Wow. Okay. You're an animal lover for sure.
Yeah, I certainly am.
So I'm looking through my notes. When we talked earlier, you talked about your work in your lab with, quote, weird mice. So talk to me about your weird mice.
Oh, yeah. So this is part of the work on sex differences. So
okay.
We have these sex differences, and how do you study that? You can do the simple thing, which is you can take away their gonads, you can castrate them or ovariectomize them, and maybe that tells you something about the sex difference, but maybe it doesn't. Another approach is an approach that was really pioneered by someone named Art Arnold at UCLA, which is to create mice that have the normal sex chromosomes. The normal sex chromosomes, women have 2 X chromosomes and men have an X and a Y. Well, what makes a male is one gene basically on the Y chromosome. So women don't have that gene and males do. So what's happened is that gene has been taken off of the Y chromosome in certain mice and put on one of the regular chromosomes that both sexes inherit. Now, when you do that, you have the possibility to create XX females, but also XX males. And also XY males and XY females. That is XY animals that have female parts, that have ovaries. And that allows you, so now you have 8 sexes. You have XX males, XX females, XY males, XY females, and then you have the ones that you've castrated. That's going to tell us a lot about the sex differences, about what's behind the sex differences. Maybe it's all the reproductive hormones. Maybe that explains everything. Maybe there's some things we haven't even guessed at yet that have to do with having testes versus having, ova. So that's that's a reason that people love our traditional animal models, because we can do such neat biology. I've got worm work going on in my lab now because I'm looking at male and female worms. We can do such neat biology. Now, how much of that is going to translate? Well, we'll see. But in the meantime, most scientists on a day-to-day basis, I have to tell you, we want to do neat science. That's what drives us.
That's great. So my final question is, and I know scientists hate to give predictions, but talk about how you see the future in the clinic. Like, let's say maybe 10 years from now. Do you think we'll be at a point where folks can go to their doctor and there will be biomarkers of aging? What might it look like, let's say, in the midterm?
Yeah, I think in the midterm there, I think we're going to have very, very different kinds of clinics, because we have lots of problems. I live in Alabama, and Alabama has a lower life expectancy than Mexico. We have health problems here that have nothing to do with living to 100 instead of living to 80. We have health problems with living to 60 or 70 or 80 years old. I think there's going to be clinics that treat these kinds of common health problems, that treat people that are obese, that treat people that don't take care of themselves. But I think there's going to also be other clinics that are going to specialize in people who are already healthy, but who want to be healthier. And, we already have these health disparities, and it would be nice if we could make these health disparities go away. But it's not going to happen. Word just came out last week on Norway, which has many fewer, it's got a lot less economic disparities than we have, but they still have this enormous range in health that goes along with education and all. So that makes me think that these things aren't going to go away. We're just going to have a medical approach for dealing with these type of people, and a medical approach to dealing with people who really have taken care of themselves and maintained themselves. They're going to need a different approach. And I almost kind of hate to predict this, because I'd like to say, well, everybody's going to just be much healthier, but I'm not feeling optimistic right now. But I think some of us are really going to be benefiting from the findings that we're coming out with right now. Maybe not everybody. It's not so much a cost thing. It's where your health problems are coming from. Your health problems are coming from incredible obesity. Then taking rapamycin is probably not what you need to worry about. Probably you need to worry about finding a way to lose some weight.
So the message is stay healthy now, so that when the therapies come, you'll actually be able to get benefit from them.
Right. Right. Something will be available in 10 years.
That's great. Okay.
Something will be there.
That's great. So thank you, Steve. This was really a great session. I want to thank you for being with us.
It was a pleasure.
I want to thank everyone who joined us. I will be back here on Tuesday, June 11th. I'll be talking with Dr. Victor Zhao, the president of the National Academy of Medicine. The National Academy is launching a global grand challenge for healthy longevity. It's a worldwide movement to increase physical, mental, and social well-being as all of us age. I'm sure we'll be talking about that. A programming note, that event will happen at 5 p.m. Eastern Daylight Time, 2 p.m. Pacific Daylight Time. So a couple hours shift from what we've been doing with our usual time frame. We will send you reminders before that event happens, and we'll also let you know when video from today's session will be ready for viewing. So a video of today's event will be posted soon. We'll let you know when it's up and available. And also a reminder, videos from our first 4 conversations already online for you to enjoy. So check out those videos. Please join me back here on June 11th and enjoy the rest of your day. Thanks for being with us.